So this is the example I will quickly go through. I'm sure you must be tired. I'm not tired. But you see, this is the kind of compounds we want for phytopharmaceuticals. See, look here. We are going by Ayurvedic traditions. We are getting knowledge from here, translating it into science, and trying to identify what are the compounds which are responsible for activity. Our crude extract was tested. Now we find that the Bacopa 1, Bacopa 2, Bacopa A, we have distinct identification of these compounds. Then Bacopa saponin C and the genin part, these are active materials, along with 2 flavonoids, apigenin, luteolin. Now, this whole material is excellent for dementia. So, someone is likely to take up this material for further in the field of dementia, because Bacopa up to the memory enhancement, other things is fine. But this is a gray area. For dementia, we don't have a drug except your glantamine, which came from Russian plant Glanthus. So these are the tests. You see how many? 23 enzyme assays. These have been tested. And receptor 3 receptors assays. Then, of course, bioactivity directed fractionations. Anti-inflammatory area, I'll go briefly through this because a lot of plants were worked by my own students. So that's why I projected this slide. And additionally, my favorite plant was Boswellia. Well, I'll give just a brief, I briefed some of the research scholars who are working. I briefed about this because when injury occurs, there is a liberation of the mediators. And those are the arachidonic acid pathways, prostaglandins, leukotrienes, and those stimulate the nociceptor, and then pain is felt. If you block these mediators by a drug, and that will take care of the pain. That is for specifically for the now this is again mechanism of action which we have given the COX-1 inhibitor and COX-2 inhibitors for the anti-inflammatory activity and lipoxygenase inhibitor. You see here the turmeric. Turmeric is acting through cyclohexane 1 and 2. Green tea also works through this area. Aspirin works through this area, goes to this route. However, ginger and boswellia, lipoxygenase inhibition working here. So very potential plants. Of course, ginger we have not pursued further, but boswellia we have gone far ahead. A lot of work has been done in collaboration with German scientists. And this, the Guwahati Pharma, we sold this, but as a phytopharmaceutical product, again, it can be developed for osteoarthritis. This is how the extraction was done. The salaki, popularly known gum resin, which is abundantly available because these plants are quite abundantly available trees. And the yield of the gum resin is quite reasonably workable, no problem getting the plant material. Then it's a non-steroidal, non-aspirin-like material which has been developed. So these are the boswellic acids which are active. These are all compounds have been identified. You see, it is a beta boswellic acid which is active. Alpha is not active. Alpha is present in very small quantity. So all these compounds have been chromatographically identified, separated, and now this is the most important ingredient of all the dietary supplements sold in the US market. Dietary supplement market is very big in US, about $22 billion market. So all pain relieving dietary supplements where there is a joint pain problems or osteoarthritis type of pain, they have a boswellia as ingredient. And boswellia has become official in the United States pharmacopoeia. So there are only 13 Ayurvedic plants which we could push forward. And since being a member of the committee of the USP expert committee, I could push forward certain plants which are very potential. So 13 plants which are worth looking for those who will be manufacturing the standardized plant extract which have a great demand. Now, this is again an example I have already cited about metformin, which has also come from the Gallica officinalis. Gallica officinalis is a Fabaceae plant, gonadin derivatives, 2 biogonadin units linked to make a metformin. So, potential antidiabetic plants, we have already started working on this. This we have finished. We are not working because industry was interested to take up the technology, but we said no, plant material is not available because the whole tree has to be uprooted. We got from forest people for research purposes 10 trees bought, and then we removed the hardwood. On the hardwood we worked, and we thought it not advisable that we should uproot the plant for the sake of this. That's why ICMR started working on an alternative. For the same segment of a pre-diabetes, we are working on the methi. Now here, these are the very potential plants for diabetes. Gymnema is potential, Chinese are working on it. Swarshya charayata, this has been worked by my students, but not for diabetes, but for other hepatoprotective formulations. And this contains the bitterest principles present in the plant kingdom. So amarygentin, amyrissurin, Swarshya-amerine. These are the 3 major compounds present besides flavonoids. So that's how this can be developed as a phytopharmaceutical. Anicostima littorale is again high concentration, very high concentration of amarygentin. This is how this was picked up. CSIR has already initiated moving forward on this plant. So here amarygentin is the main, and beside this there is a swarcetin. Swarcetin is a C glycoside present in this, which is quite active and stable and druggable. And then, of course, other vitexine, isovitexine are present. So this would go as a phytopharmaceutical route. Salicia prenoides, this was worked long back. At a time when the chemists were not interested in looking into the aqueous materials. But these are the compounds which are active. Murraya cuenga is another plant which has not yet been touched, but worth looking into it. And Murraya cuenga is leaf. This is kadi patta, very important plant to work with. Well, these are some of the compounds which have been identified in Pterocarpus marsupium. I'm sure most of you are tired, so I'll go. This is just a extraction and fractionation procedure which we have used for the present antidiabetic. So what does it contain? It contains pregnenolone, which is alkaloid. It contains 6-hydroxyisoleucine, which is amino acid. It contains the protodiosine derivatives, trigoneosides. Those are the most potential compound of the saponins. And these are the compound that we are using as the markers, as the phyto. So this work is pursuing very fast. Liver protecting, of course, this was at one point of time, it was very area very of great interest to me. But subsequently, I shifted from liver protecting to other areas. But a lot of plants have been identified. You would find a book written liver is such an organ where all the metabolism taking place, all xenobiotics or our materials that we consume, xenobiotics, they metabolize in the liver. So liver is such a sensitive organ. The protection of liver is extremely important, but nothing is available in the modern system of medicine where the liver can be protected. Nowadays, fibrotic liver is a serious issue. So what we are trying to position, the Picroiza curua as one of the candidates for taking it forward for fibrotic liver. How much success we'll get, we do not know. But because of the background of the Picroiza curua and Endocrine paniculata, which are very good hepatoprotective in India, and we are trying to pursue this for fibrotic liver, which is again a big challenge in the modern drug therapy. Well, this was the book which I wrote round back in 2008 or 6, 2006, and This was published by ICMR under their program. This would contain different testing methods, and 12 plants have been covered which are most potential for liver protection. And then what is the concept of these plants, used these plants in the traditional system of medicine? How comparable in the modern system of medicine? What is the treatment available in the modern and traditional, both Ayurvedic and Unani? That kind of comparison has been done in this book, worth reading if you get some time. Icorizacuru, of course, these are the erudite glycosides, which are the potential bioactive compounds present in it. Well, Endographis paniculata has been positioned in India for liver, but in China, when I was with Glaxo, they were very keen to position this drug on the Chinese pattern for as a calm cold drug. But they abandoned the idea later on because there were some issues of the pesticide residue which was coming in the extract of the Endographis paniculata. But this is a plant material. If we follow the good agricultural practice to cultivate, because it's an annual plant, no problem of cultivation, and rest assured that there's no contamination of the pesticide, because those pesticides probably creep when it's collected from wild. From the agricultural crops, whatever pesticide they use, they creep to the soil and they contaminate the medicinal plants. This is how it should be avoided. Anyway, this China is using for, Thailand is using for cough and cold as a mild treatment. So it may be good idea to pursue it because all the compounds have been identified as a endographolide, neoendographolide, isoendographolide, endographenine, endographoside. All these compounds have been identified. Well, ossimum, our day-to-day use plant material, every house garden would have Tulsi in the house, but very promising plant as an anti-stress agent, anti-stress, anti-inflammatory, and the compounds which have been identified are the triterpenes like the olenolic acid, rosmarinic acid, oxalic acid, and eugenol. Besides this, these are the two new compounds which have been isolated from ossimum. So ossimum is becoming very popular as a under the dietary supplement in the US. Dried leaves are being sold on the shops at a very high price. So, of course, all good agriculture practices followed for cultivating, for drying, and those are packed nicely, and you get $5, just 10 gram of Tulsi leaves. So people buy it. Well, the data which is required, it's a long journey now. If I explain each and everything, probably it will take a lot of time. But when you reach that stage of developing a dossier, these are the things one has to take care of. Your identification of the plant, correct identification of the plant, its nomenclature, and origin of the plant, traceability of the plant material that you get, whether you get from wild source, which source, what is the geographical location where the plant was growing. All these information have to be given. It's full authentication following all the procedures, macroscopy, microscopy, all parameters that I showed in the earlier slide. Process of extraction and subsequent fractionation, the way I have shown, in the same way it should be done. Then formulation of the phytopharmaceutical, how you make the formulation, what excipients you use, manufacturing process. Now this manufacturing process has to be done at a GMP approved lab. On 5th October 2018, the Drug Control General issued a revised Schedule M of the good manufacturing practices in the modern medicine, where chapter 6, part 6 of this schedule contains the GMP for phytopharmaceuticals. Then of course, stability data has to be generated. So I'll quickly pass through these slides. Formulation, stability data, of course, 40-degree temperature, and you have to monitor every month for 6 months, humidity control. So the stability chambers are available, so everything is set. You have to keep your sample. One sample without the pack, one sample with the pack that goes to the market. So the safety, of course, is a stringent rule for safety evaluation. Then GMP to be followed. Human studies, of course, phase 1, phase 2, phase 3, all these things have to be followed. So every step you have to take permission. For first, you take permission of the Drug Control General of India by applying. The fee is 50,000 rupees for first application, and then followed by 25,000 for subsequent second and third clinical trials. Then, of course, the clearance comes after complete review. If they're satisfied, thereafter, you have to have a marketing information. How you market, what are the pamphlet that you are inserting into it. Then post-marketing surveillance. For first 2 years, every 6 months, you have to submit your records, where you have sold that drug, how it is behaving in the patients. For first 2 years, your drug will be monitored. If there is any toxicity side effect, the drug would be withdrawn. After 2 years, then of course, at any time, DCI can ask the sample for verification. I think that stops me. This is all the checklist I have given. I will not narrate again. This is for the benefit of those who want to submit the dossier. So this is all from the, in fact, the Gazette notification that I have listed. Well, the disease priority area, that 21st century, what are the diseases? That's the last slide which I am projecting. The protozoal diseases, of course, malaria, filaria. Of course, we are getting away from it, but we have to take care of still the malaria and filaria. Of course, viral diseases, big alarm, sometimes become an epidemic, and viruses, they change their profile because of the mutation taking place. So we have to be extra cautious when developing drug for the viral diseases. And initiation has been done by some of the DBT labs to take initiative, because ICGB has done good work in the dengue. But we want that some institutes should take up at national level the antiviral activity in a big way, because viral is a real problem. Then metabolic disorders, of course, already your people are working, but be careful where to divert, where to stop, and where to go ahead. Then disease of complex etiology, I think we should not touch at this stage. We should not get into cancer. We should not get into the muscular dystrophy, Parkinsonism. These areas should be avoided for institute of this stature. Cardiovascular, CNS. Cardiovascular, again, coronary vascular disease is important, but again, plethora of drugs are available. Better not to touch it. But CNS, yes. CNS, not much drugs are available. CNS area is worth taking forward. Then self-inflicted disease, same metabolic syndrome, your obesity and other things. So I think that's what I could explain. I'm sorry troubling you so much, but I thought at the appropriate time, so kindly given by you, to a person like me who is out of research, totally, I am away from research. I'm not doing any active research, but it is always a pleasure to speak on research, to talk on research, to explain the experience that we have gained, and our young generation should come, should make the name of the country. And that's our ambition, that our natural resources should be properly utilized, and persons like you can think of the valuables. And one scientist, if works sincerely, comes out with product, how many people will be benefited? Besides the patient, how many people will be benefited? The cultivators, the processors, the manufacturers, the others. So this is a great service that a researcher in the drug discovery and development, a person must be considered as a real sannyasi, karam yogi, who reached to a level of producing a drug. Unfortunately, I attempted, I failed. I could not produce even a single drug to that level. But I expect if any one of you comes out with that level of research, I'll be very, very happy. Thank you.